The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Viral Clearance, Removal of Viral Contamination in Biopharmaceutical Manufacturing. Now, if you miss one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by following Nelson Labs on LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There, you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for consumer care, medtech, and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and personal care product industries. The experts at Nelson know every test matters and requires solution to complex problems to improve patient outcomes and minimize client risk. And now, let's get started. Today, we're joined by Kelly Burningham. Kelly is a highly qualified virologist with a degree in microbiology from Montana State University. With significant prior laboratory experience, she joins the Nelson Labs team, formerly known as Bioscience Laboratories, in 2007. Currently, Kelly serves as a Study Director 3 at the Virology Laboratory at our Bozeman facility, where she plays an instrumental role in offering expert guidance to our sponsors on virus testing requirements. We do encourage your questions. Submit them at any time in the question box. Kelly will try to answer as many as she can in the last 10 to 15 minutes of the webinar. And now, beginning today's presentation, I turn the time over to Kelly.
Good morning or afternoon, depending on where you are joining from. And thank you for the introduction, Daniel. Today, I will be talking about an overview of the importance of viral safety for biopharmaceutical products, and I'll be discussing the steps of a viral clearance study and considerations to take into account when developing your manufacturing process. So here you'll see the overview of the presentation. Today, we'll talk about an overview of what biopharmaceuticals are. We'll discuss the viral safety importance and some of the regulations. And the next three, the viral clearance process validation, GMP screening, and disinfectant validation are all critical parts of a viral safety program. So what is a biopharmaceutical? So the FDA characterizes biopharmaceuticals as pharmaceutical products composed of sugars, proteins, or nucleic acids, or complex combinations of these substances, or maybe living entities such as cells and tissue. So most of what we're going to talk about today are tissues and products that are derived from human or animal tissue. They can be prepared for many uses. Some of the examples include skin grafts, collagen injections, bone substitutes, also vaccine production. And then now we'll discuss viral safety importance. So because biopharmaceuticals and biologicals are derived from human and animal tissue, they are at risk for virus contamination. The contamination can either come from the cells themselves or from introduction of adventitious viruses during the manufacturing process. An example would be reagents such as FBS, or there can be environmental contaminants from the facility or the process itself. Viruses are more difficult to detect than other microorganisms, which is also an issue with viral safety, so they're a little bit more difficult to detect. So far, there haven't been any cases where virus has been detected in the final product to be able to infect humans. We will all remember back in the in the 80s when some of the blood plasma was spreading HIV amongst patients. That was a different type of product than what we're discussing today. But one instance that can happen is bioreactor contaminations have occurred with adventitious viruses, and that can cause a huge financial risk to the company. There will be investigations, the facility will shut down, there will be market share loss, and the customer will have a loss of confidence in the company. The risk to patients can be direct infection risk, adverse reaction risk, reduced efficacy of the product, and then also with the facility having to shut down, there can be drug shortages for people who need them. And because of all these issues, viral safety regulations were put into place to mitigate these risks. So these are some of the viral safety regulations. There's the ICH-Q5A that is currently being revised. It's from 1998, so as you can imagine, there's a lot of new technologies and emerging therapies that have come through since 1998. So right now it's in the draft procedure, and I think it's supposed to come through next year. So there's also the ISO medical device, and then the European agency regulations that give you guidances on how to test. Oh, excuse me. So there's three major considerations or pillars for viral contamination strategy, and it helps to show confidence in the safety of your product. So the first one is to select and test raw materials and media components for virus contaminants, so you know what you're starting with, any known viruses that you can detect, and excuse me, just a starting point. And then the second one is the assessment of your manufacturing process for the ability to inactivate and remove viruses. This is going to be your viral clearance validation study that we will talk more in depth later on in this presentation. And then the third is to test the product for contamination at appropriate steps during the manufacturing process. So the number one is more upstream, and then number three is more downstream through your manufacturing process. So now we will move on to discuss viral clearance validations. So viral clearance validations are to assess the manufacturing process for its ability to inactivate and remove virus contaminants. So inactivation is reduction of virus infectivity caused by chemical or physical treatment, and removal is the physical separation of virus particles from the intended product. Viral clearance studies help to ensure that your biopharmaceuticals will be free from virus contaminants. Excuse me. Each end product and manufacturing process is individual and it's case by case. So each starting material is going to have, there will be many different starting materials from animal or human tissue, and each process will just be individual for that company and for that product. So this is a really big area. And we always recommend to consult your regulatory agency at the beginning of this process for your individual needs for your product. So the objectives of viral clearance are to demonstrate clearance of virus known to be present in the animal-derived material, to ensure potential adventitious virus introduced during production will be cleared, and to assess the critical steps of the process to effectively inactivate and remove virus. And then the end goal is to estimate the overall reduction in virus titer. So this slide shows the general steps of a viral clearance validation study. First, you'll start with a risk assessment of viral safety of the product, target virus selection, selection of critical processing steps, scale down, determine viral interference and cytotoxicity. Then we'll discuss the viral clearance study and then calculating log reductions. So the first general step is the risk assessment of the product. So you want to perform a risk assessment to find potential sources of viral contaminants. So either from virus found in the original source or viruses that can be introduced during the manufacturing process. This can be literature reviews, screening tests through in vitro cell culture tests, electron microscopy, and PCR screening. And you'll want to, that's so you'll have knowledge of how much virus is present in your material or in your procedure. So the next step is target virus selection. So where you'll choose the virus, you'll want to choose viruses that represent a wide range of physiochemical properties. So we'll have and some examples of categories to choose from would be DNA, RNA, enveloped, non-enveloped. And the ICHQ5A defines these viruses as the following. So there is relevant viruses, which are identified virus of the same species that is known or likely to contaminate the cell substrate or any other reagents or materials used in the production process. Then there's the specific model viruses, which are viruses that are closely related to the known or suspected virus, and they have similar physical and chemical properties. Then there's the non-specific model viruses, which are the viruses that are used to characterize the robustness of the process, how effective it is for a broader range of viruses. You also want to choose at least one highly resistant virus to include in your study. One example would be porcine parvovirus. It's resistant to a lot of different chemicals, and it'll show how robust your procedure is. You'll also choose a virus that can be grown to a high titer. You also want to choose a virus that there's an efficient and reliable detection in the laboratory. So it'll be a virus that can produce good cytopathic effect in the host cells, or you're able to effectively detect it so you can get accurate results. Then you also want to make sure that it's safe, the viruses are safe and feasible for your testing laboratory. So the next is the critical step selection. So more than one step should be considered. The minimum is 2, but most commonly 3 to 5 are tested. The critical steps should be individually assessed, and you want to choose steps that are known to be able to inactivate your target viruses. So some examples are low or high pH, detergent treatments, heat treatments. The next that we'll talk about are, you want to test intermediate materials. So at each critical step, you want to test that specific step from start to finish. So you'll spike the material, it'll go through that part of the process, and then you'll see the log reduction from start to finish. You also want to consider worst case conditions. So an example would be the lower end of your pH range or the lower end of your time point, just so you can ensure that your process is clearing the virus. Kinetics to make an activation curve are also required. So you want to choose at least 2 time points for each of your critical steps. One time point is you want at least 1 time point less. You want 1 time point less than the minimum exposure time, but greater than 0. And then you want your minimum exposure time. You also want to test the same buffers and reagents that are used in the commercial process, so there's less variation. The process should also be followed as closely to the commercial process as possible to ensure that you're actually testing how valid your procedure is. So the next is the scaling down. So most manufacturing processes are large volume, full scale. The testing facility may not have the capability or the capacity to be able to perform that testing there. And also to prevent introducing unintended virus into your production facility, viral clearance studies should be tested in a separate experienced virology lab. And other reasons that the testing lab will perform scaled down testing is, as I said earlier, more feasible for the facility and to reduce the amount of virus needed for the validation. The validity of the scale down should be demonstrated, excuse me, and it should represent the full scale manufacturing process as closely as possible. So things to take into consideration when you do scale down your procedure is you want to keep, the buffers and the reagents as similar as possible, the pH and the temperature. You want to make sure that the protein concentrations, there isn't much of a change or much of a difference. And the product should be representative of the full commercial scale manufacturing procedure, which would be the intermediate steps at the different critical steps in your manufacturing process. So viral interference and cytotoxicity controls are necessary controls to add to your study. And that's because some production components and the intermediates themselves can interfere with the ability of the virus to infect the host cells. And also this can cause an adverse effect on the host cells used for virus detection. So controls are typically performed prior to your viral clearance validation, so you can show if there is any interference in cytotoxicity, and if there is, the validation can be modified to remedy that. So now we'll get into the actual viral clearance portion of the study. So intermediate materials from each of the critical steps that you chose before are spiked with a known titer of virus and processed through your procedure. So the number of replicates, the ISO and ICH state that testing should be performed with a sufficient number of replicates and controls to ensure statistical accuracy of the results. We recommend 3 as your minimum. And each critical step needs to be followed as closely to the commercial full-scale procedure as physically possible. It's very important, as I'm sure you've noticed. And then after exposure has been completed, the virus is collected, diluted, and titrated to determine the amount of inactivation. You'll also have controls evaluating the recovery of the pre-treatment virus load, so you can determine the log reduction. We call this an input control to show your untreated, it's kind of like a baseline or the amount of virus that you input into the study. And we also perform PCR on samples to show removal of the virus from your procedure. So the last step is to determine the log reduction values. So viral recoveries from the untreated materials are compared to recoveries at the end of the processing step to determine the log reduction values of the clearance steps. So the log reduction value from each process is added, and you can add together to obtain the viral clearance value at the end of the study. An effective viral clearance step for it shows a log reduction of 4 logs or more. A log reduction of 1 or less is considered negligible. We recommend contacting a regulatory agency to find out, for you to find out what your necessary log reduction value is for for your specific product type and your manufacturing process. They can help you figure out exactly what they do, what they require you to need, and it all pretty much depends on your regulatory auditor. So now we'll move on to the GMP screening and disinfectant efficacy. So you'll remember this slide from before. So this will be the screening of the raw materials and media components, so your unprocessed bulk, and then also screening for virus contamination at the appropriate steps during your process. So some GMP screening considerations are, so you'll screen to qualify your source cell lines or your unprocessed bulk materials, and then you can screen the media components used in the process to show what you're starting with, and also what you don't want to use in your manufacturing process. There's different types of testing. There's in vitro methods, there's cell-based assays, electron microscopy are two examples. There's in vivo methods, which would be antibody detection in animals. These will be explained in more detail at a future webinar. What we're going to talk about today specifically is the molecular methods, so PCR. And so our capability is our PCR, we can detect known viruses or closely related virus families. You kind of need to know what you're looking for this type of detection, and that isn't always possible. There is next generation sequencing that has the ability for broad virus detection, and we are hoping to be able to offer this in the future. So screening should be done at upstream and downstream parts of your procedure to be able to show what you're starting with, like I said, and if you were able to clear or if there was contamination brought in at different processes during your procedure. So some applications of GMP screening are for lot release, quality control, contamination control. And this is a service that we offer here at Nielsen Labs Bozeman. Then we'll go into the disinfectant validation. So disinfectant validations are important because environmental contaminants can be introduced during any time in the manufacturing process. It can happen from employees. It can happen, from different surfaces in the facility. So you want to make sure that you have an effective cleaning and disinfecting procedure for the manufacturing facilities, so you can show product and employee safety. So when we perform these validation studies, we'll test different surface types from your facility. Some examples are stainless steel, glass, vinyl. We'll contaminate those with either the bacteria, fungus, or virus, allow it to dry, and then we'll treat them with your in-house cleaning procedure. So whatever disinfectant or chemical sterilant, that process, so you can show that your disinfectant procedure is actually killing any contaminants from bacteria or fungus. Also, if you work with viral vectors on helper virus, it can show that you're able to inactivate the helper virus. And then another example would be blood contact surfaces. So this concludes my presentation. I would like to thank Daniel for his assistance, and we are open to any questions you may have. Thank you.
Kelly, thank you so much for your insights. We've all learned some great information today, and you've given us a lot to think about. And we did receive some questions as you spoke today, which we would like you to address now. Before we get into those, I just want to let you know that if we don't get to your specific question, Kelly will be happy to address it individually after we conclude today. With that being said, we'll jump into the first question here. And the question is, how do you select the viruses used in a viral clearance validation?
So viruses are usually selected based on what your starting material is. So you could do a literature review and find out what types of viruses can infect that actual material. Also, you can do the screening validations, well, not validations, but the screening test to see what you're starting with. You can also see what contaminants can be introduced from your facility. So basically, that information will kind of tell you how to select. It's also if what your endpoint you want is. If you want to have a really robust procedure, you want to use a broad range of viruses that are known that could potentially infect your starting material. You also would want, as we said before, you'd want a non-enveloped DNA, a non-enveloped RNA, an enveloped DNA, and enveloped RNA, just kind of a broader range so you can show that your clearance can broadly inactivate or remove and that your product is essentially safe.
Thank you so much. And okay, so now the second question we received is a 4-log reduction the minimum value for viral clearance?
So the log reduction, you would like to have a 4-log reduction from each critical step. We recommend to have at least a 6-log reduction for your cumulative value. But however, it depends on the viral load in your material, in your starting material. It can also be yeah, and so yeah, so a 6-log reduction, my apologies, is what we recommend. It's a good starting number to show a robust clearance study. However, you do want to reach out to the regulatory body and they can tell you what you should expect. So so there really isn't a definitive answer, but a 4-log reduction definitely shows a good reduction.
Thank you so much. The next question we received was, is the viral clearance process the same for other biologics, such as a tissue product?
So the viral clearance study it'll be specific for your starting product. And then you go through each critical part of the manufacturing process. So it's essentially the same idea, but each study will be different because your starting material is different, the manufacturing process is different, and that needs to be followed as closely as possible to show the efficacy of your process and the safety.
Fantastic. Thank you so much. The next question we received was, can you perform the same validation against other pathogens like bacteria and fungi?
You can. This would be different guidances because bacteria and fungus, the way that they interact with tissues is different. But yes, there are tests out there. There's regulations for chemical sterilants and other processes to show that you removed any bacteria and fungal contamination.
Kelly, thank you so much. And the final question we have time to answer today is, does your viral removal approach, such as chromatography or radiation, affect how you would conduct a viral clearance validation study?
It won't necessarily affect how you run your study. You'll want to follow the process as closely to the manufacturing process as possible. So the radiation exposure, your chromatography, you would want to make sure your scaled down approach is very similar to the commercial manufacturing process. And So basically just follow the manufacturing process as much as possible. So that's how they're kind of all similar, is that you just follow the process as best as possible.
Kelly, thank you so much. Unfortunately, that is all the time we have for questions today. If you submitted a question and we were not able to answer it in this session, our experts will try to provide an answer via email after the conclusion of this presentation. You can also contact us directly at our email, experts@nelsonlabs.com. I'd like to thank our presenter, Kelly, for their insights, and I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience. Thank you, everyone, for attending, and we hope you have a wonderful rest of your day.